Bismillahirrahmanirrahim. Dear students and audience, in this video we will talk about population genetics and Hardy Weinberg Equilibrium or law. Population genetics is the study of genetic variation within and among individuals. Alternatively, it is the study of factors affecting gene and genotypic frequency in a population. So let's define what population is. It is totality of individuals, or it is a group of individuals of the same species that live in the same geographical area and are capable of interbreeding. For example, Holstein cattle of Canada, Neely Ravi buffalo of Pakistan. How we can define a gene? It is basically a region of DNA coding either for the messenger RNA encoding the amino acid sequence in a polypeptide chain or for a functional RNA molecule. And what is the genotype? It is the genetic makeup of an organism. It describes an organism's complete set of genes. So the process of determining a genotype is called genotype. And another relative term is allele. Allele is a variant form of a gene. For example, humans are diploid organisms because they have two alleles at each genetic locus with one allele inherited from each parent. So each pair of allele represents the genotype of a specific gene. Now, what is locus? A locus refers to the specific location of a gene on a chromosome. So it is a home address of gene on a chromosome. It is called locus. For example, let's have an example of a typical gene. For example, in cattle, we have a bovine beta-casein gene, which is called CSN2 gene. It is a protein coding gene, and its locus is chromosome number 6, and it has two alleles. Most common ones, there are many alleles, but most common ones are A1 and A2. A1 is considered bad one, and A2 is considered good one. And based on these two alleles, the cattle have three genotypes. A1, they produce bad protein in milk. A1, A2, they produce mixed protein in milk, and A2, they release good protein in milk. And a relative term also is haplotype. A haplotype is a group of genes within an organism that was inherited together from a single parent. It is a group of genes within an organism that was inherited together from a single parent. It is haplotype. So now let's define allele frequency. It is the frequency of a particular allele in a population. Allele frequencies can be represented as decimal, 0.01, or percentage, 1%, or fraction 1/100. So all three forms can be used to represent frequencies. For instance, frequency of A1 allele of the same CSN2 gene in cattle and frequency of A2 allele of CSN2 gene in cattle. And let's look at genotypic frequency. It is the relative frequency of a particular one locus genotype in a population. For example, frequency of A1 genotype for CSN2 gene in cattle, frequency of A1 genotype for CSN2 gene in cattle, and frequency of A2 genotype. So we can describe cattle in three different genotypic forms, and we can calculate their frequency. So Hardy Weinberg Equilibrium was established in 1908 when Hardy, a British mathematician, and Weinberg, a German medical doctor, they independently proposed this principle of population genetics, which is now commonly named as Hardy Weinberg Equilibrium. It states that in a large random mating population, in the absence of selection, mutation, and migration, gene and genotypic frequencies remain constant from generation to generation. So large means population would be in hundreds, and random mating means every male has equal opportunity of mating with every other female, and every female has equal probability of mating with every other male. So it also says that genotypic frequencies are related to gene frequencies. For example, gene frequencies in parents are P plus Q square, and genotypic frequencies in progeny will be equal to P square plus 2PQ plus Q square. And in the same way, capital P, which is genotypic frequency of dominant homozygous dominant genotype, it would be equal to small p square. Frequency of heterozygotes H would be equal to 2pq, and frequency of homozygous recessive genotype would be equal to small q square. Capital Q is equal to small q square. So here I have defined the terms. P is the frequency of dominant allele, for example A. Q is the frequency of recessive allele, small a. And capital P is equal to small p square, which is the frequency of homozygous dominant genotype AA. Capital Q is equal to small q square, which represents the frequency of homozygous recessive genotype small a. And H is equal to 2pq, which represents the frequency of heterozygous genotype capital A small a. So, additionally, the sum of allele frequencies for all the alleles at one locus must be 1. So, therefore, Hardy Weinberg equilibrium says P plus Q would always be 1, and similarly, P plus Q plus H is equal to 1. If the P and Q allele frequencies are known, then the frequency of three genotypes can be calculated using Hardy Weinberg equilibrium. So we only need to know one of them, either P or Q, then we can determine the other one. And once we have these two, we can calculate genotypic frequencies from these formulas. Capital P is equal to small p square, capital H is equal to 2pq, and capital Q is equal to small q. So in population genetic studies, Hardy Weinberg equilibrium can be used to measure whether the observed genotype frequency in a population differ from the frequencies predicted by Hardy Weinberg equation, and usually we use a chi-square test to measure it. So no population may be seen in Hardy Weinberg equilibrium in reality, but some may be close to achieve Hardy Weinberg equilibrium. So now we have described the Hardy Weinberg equilibrium. Let's look at the forces which change gene and genotypic frequency in a population. Mutation is a process that alters DNA to create new alleles, but mutations are rare events. 10 raised power minus 7 to 10 raised power minus 8 per nucleotide per cell division. So therefore their effect could be small, but mutation can always increase genetic diversity within a population. Another force is migration, which is movement of into or out of a population. Introduction of a large number of genetically different individuals in a population would have large effect, and migration essentially increases genetic diversity within a population. Selection is another force, which is a process in which certain individuals are preferred over others for the production of next generation. Natural selection is selection by nature, and artificial selection is selection by man. And selection can decrease genetic diversity. Within a population. And another force is a random genetic drift. It is change in allele frequency of a small population due purely to chance, and it can be observed in two cases: when individuals have been reduced due to a natural disaster, it is called bottleneck effect, and when number of individuals separate from their main population to form a new colony, which is founder effect. In both these cases, random genetic drift becomes important. It can result in loss of a allele, or it can result in fixation, where only one allele becomes the only allele at that locus. Genetic drift is unpredictable. It is like flipping of a coin. We do not know which way it will get. And random drift decreases genetic diversity of a population. Thanks for your attention. If you have any question and comments, you can put it in feedback. Again, I'm thankful for your attention. Stay blessed always.